Welcome back to part 2 of our discussion of drug discovery and development. In the first module, we talked about drug discovery and how we learn about diseases and drug targets and develop drugs and do preclinical testing. And if those lead compounds, as we described them in part 1, if they are optimized and developed in a way and evaluated in preclinical trials that they have promise, they might go into clinical trials. And that's what we're going to discuss in part 2, drug discovery and development as it relates to clinical trials, the development process. And this use of the development process is really more of an evaluation process to determine if this compound has promise for real use in patients with the conditions that we're interested in treating. This slide kind of reiterates kind of the overall process that we're going to be talking about. So we talked about in part 1 that we have a drug discovery in that first arrow, and that if that compound continues to show promise in its preclinical testing, and we can optimize this molecule and develop it in a way that seems like it has promise for clinical use, that company or that institution that has developed this product would apply for what's called an investigational new drug application, IND, with the FDA. And this is to allow testing in human beings. So an IND is filed with the FDA that allows us to go ahead and do some clinical trial work before it is brought to FDA for final approval. In that process, we're going to use a thing that's called an IRB, or Institutional Review Board. That's one of our pharmacy fundamentals for today as well. And we'll describe this IRB as there to help protect human subjects that are undergoing investigational drug studies, or any kind of research for that matter, to make sure that they are protected, that they are giving informed consent, that they understand that they can voluntarily withdraw at any time, and that the benefits outweigh the risks to these human beings who will be participating in this study. And once this IND has been filed and approved, and once the IRB does their work, then we might move this drug into clinical trials, which will be a very significant part of this drug development process. This slide kind of outlays that entire process for you. We're going to talk about 3 phases of this clinical trial kind of continuum, if you will, phases 1, 2, and 3. And if those are successful, we would eventually then go to send this drug to FDA approval. And we'll talk about that process in our next video module. So let's take these clinical trials one by one and think about them for just a few minutes. First of all, I want to think about phase 1. This is the very first use of this new compound in human beings. Typically, we have fewer than 100 healthy volunteers who are going to try taking this drug. These are not people who have the condition we wish to treat. We're just simply going to try this in human beings to think about developing the dosage better, to discover any safety issues which might emerge with these healthy volunteers, and to start looking for side effects. And really, all of these things, dosage, safety, side effects, will be screened as we continue through all of these phases and eventually try to determine if this is safe and effective to use in real patients. Phase 1, if that's successful, we would continue to phase 2. And in phase 2, we start to use patients that have the actual condition that we are hoping to treat, usually about 100 to 500 of these patients. And then now we're going to start looking at efficacy, or is this compound effective in treating the condition that we think it's going to be effective in. And also we'll continue to look at safety and dosing, as I said before, but we're going to start to take a look now about efficacy in particular. And quite frankly, many compounds stop at phase 2 because we start to find out that this drug is not going to do what we think it will, and there's no reason to go on. If we go on, we go to phase 3, and this is the most expensive part of the process. So there's a lot of really critical thinking that happens in phases 1 and 2 before we go to phase 3, because now we're using thousands of patients, usually in multiple sites with the conditions of interest. We're going to be further testing whether this drug is efficacious. We're going to be looking for adverse drug events. And in this phase 3, we're typically going to be using randomized controlled trials. This means we're going to be comparing this new compound to maybe a placebo. There's a pharmacy fundamental that we'll learn about in a few minutes, or maybe other drug therapies, and it's typically done in a double-blinded fashion, meaning the patient nor the person who is treating the patient knows which compound that person is getting, whether it's a placebo, which means that there's no drug content in it, or whether it's the drug or another drug therapy that's being compared with this new compound. So that way, the patient doesn't have expectations that might affect their behavior or affect even their outcomes, and the person who's treating the patient is not biased as well. This is a very expensive part of this process. So, those compounds that go to phase 3 have been thoroughly vetted before they get to that point. And if we think about the clinical drug funnel, we mentioned in our last video module that we might start with 5,000 to 10,000 compounds as potential lead compounds early on in this process, that by the time we go to clinical testing, we've narrowed that down to maybe 250 compounds. By the time we go to clinical trials, as we can see here, we narrow that down to about 5 compounds. And of those 5 who go to clinical trials, typically only about 1 will make it to the market. So you can see kind of the risk here. We start with maybe 10,000 compounds that we think might have promise. That narrows down to maybe 1 going to market. We start to understand why this is such an expensive process and why maybe drug research expenditures are so high, and maybe start to understand a little bit why maybe drug compounds are so expensive on the market. This process of clinical trials is also a lengthy process. As we mentioned that preclinical testing usually lasts from 3 to 6 years. Clinical trials timeline is typically more like 6 to 7. So we're investing a lot of time, a lot of money, a lot of effort, a lot of scientific expertise in this process. Let's look at our pharmacy fundamentals for this module. Drug development is the process through which a new drug candidate is optimized, formulated, and tested for use in humans. An investigational drug is what we're using throughout this process. So when we enter phases 1, 2, and 3, we are using drugs that we're now going to be calling investigational drugs. And these are new medicinal agents being tested in clinical trials for potential FDA approval, an investigational drug. I mentioned to you already this idea of an institutional review board, or IRB, being important because they must review and approve all research involving human beings. IRBs help ensure that the risk to benefit ratio for patient involvement in research is reasonable, that patients are fully informed of all parameters of the study being conducted, that patient involvement is voluntary with signed informed consent, and we'll talk about that in the next slide, and that patients may voluntarily withdraw from the study at any time without penalty. Really critical part of this process. Informed consent, that is a big part of the IRB process, is informed and voluntary agreement for treatment made by a competent patient or research subject. Think about informed consent in terms of research, but I also like to think about informed consent in terms of just our standard medical treatment. Shouldn't we give patients good information about our medication therapy treatments that are about to begin, and so that they can voluntarily agree to participate in this medication therapy or not? Randomized controlled trials we've described as being used in phase 3 trials, and these are used to evaluate the efficacy of a new drug candidate against a placebo or other medications. These trials are typically blinded so that neither the patient nor treating investigator is aware of what the patient is receiving. And a placebo is an inert material designed to look exactly like an actual drug product. A placebo effect is an improvement in a patient's condition which cannot be attributed to medication or treatment with active ingredients being provided. Well, here's my discussion forum question for this module. Why is it so important to have IRB review of research using investigational drugs in patients? I'd like to hear what you had to say about that on our discussion forums this week. And we'll see you next time when we kind of complete this cycle and think about what do we do with these compounds when we go for FDA approval.